Because active transport does not depend on diffusion, cells can use this process to concentrate molecules within the cell or to remove waste from a cell. And it's going to move against the concentration gradient. It's going to go from low concentration to high concentration. So you're going to actually pump those molecules using proteins and energy, usually in the form of ATP, across the membrane from low to high. Calcium, potassium, sodium are all molecules that must be forced across the cell membrane using active transport. So active transport moves molecules up a concentration gradient, generating potential energy across a membrane. That energy must come from somewhere. So active transport converts energy stored in chemical bonds into energy stored in a concentration gradient. When ATP reacts with the transport protein, the energy stored in that phosphate bond is released. The reaction of the phosphate with the transport protein changes the shape of the transport protein, moving the molecule across the membrane. The energy is therefore transferred from the ATP to the transport protein. And the proteins, like the example here for ATP energy, the sodium-potassium pump, are often called ATPases because they use ATP. But ATP is not the only source of energy in primary active transport. There's also redox energy, like you'd see in the electron transport chain in mitochondria, and photon energy, like you see in thylakoid transport in the chloroplast. So in primary active transport, the major differences here are the type of energy. In primary active transport, you used energy directly to make transfer happen. But secondary active transport is a bit different. Basically, in this one, you use the energy of an electrochemical gradient. As ions pass back and forth across a membrane, a charge is built up. Same way when you have an electromagnet or a motor or a generator, when that magnet is spinning or when the parts are spinning, it tears off electrons and you generate a charge. This generates power in the form of electrical energy. Now, it can be symport, as we can see in the top, where the sodium and the glucose are being transported together, or it can be antiport, where the passing of one allows the passage of the other. So the ion is traveling down its concentration gradient. It's a passive process, but the power that it generates allows the transfer of something that would not normally be able to transfer. So just like in primary active transport, the proteins undergo a conformational change. They change their shape. Now, this shape is not always changed just by this process. Some of them require other molecules to trigger it. An ion-gated channel will only open when a certain ion is bound. A voltage-gated channel will only open when the membrane is depolarized enough and there's enough of a charge difference to make that process happen. If you're dealing with a lot of material or very large molecules, you can't use a protein to do it because sometimes the amount that you're looking at is even larger than the protein itself. Say, for example, you need tons of glucose or something. You're going to need to use a larger process, and that process is called bulk transport. So it's all about vesicles. As you can see on the right, the material that needs to come in is grabbed in a vesicle and it comes in through endocytosis. When it needs to leave the waste products or whatever, it binds with the membrane and allows it to exit, or exocytosis. There are three major types of endocytosis. Phagocytosis is called cellular eating, and it's going to bring in large amounts of solid substances, but it only occurs periodically and only occurs in specialized cells, for example, the white blood cell. So when a white blood cell is engulfing an invader, it's phagocytosis. Now, penocytosis is taking in liquids and dissolved materials, and all cells are capable of this. It's very small amounts. It's often called cellular drinking because you're looking at the liquids versus phagocytosis looking at the solids. Now, both of those are very non-specific. They grab whatever's there. Receptor-mediated endocytosis is super specific, and it requires the trigger of a particular signal molecule called a ligand. A ligand is any molecule that triggers a signal. It's just a general word. It's very, very efficient, and it only occurs for certain things that your body really needs. But unfortunately, it's also trickable, and this is how some viruses get into the cells. Now there's actually only one type of exocytosis, and this is just when the vesicle binds with the cell membrane, fuses and releases the material into the outside. That vesicle becomes part of the cell membrane and the material exits. So it's fairly straightforward.